Welcome everybody to another Beyond Terrain mini lecture. Today we are going to be discussing the fact that viruses have never been demonstrated to exist. I know this has been discussed ad nauseum already. There have been some great minds out there. I thought I'd put together just a little quick, concise video on the topic, so hopefully this doesn't drag out for too long. Before we get started, if you're interested in implementing terrain principles into your life, how do we operate within such a framework where microbes don't cause disease, viruses have never been demonstrated to exist, we can't rely on these modern methods for understanding things through these empirical false observations without considering falsification. That's what the Beyond Terrain community is for, so make sure you go join that waitlist right now. A place where like-minded individuals are going to come together. We're going to dive way deeper into terrain principles, how to implement them into your life, and so much more. So go over there, check it out, join the waitlist. You won't want to miss it. So the mainstream looks at viruses as being self-replicating particles that cause disease. The idea of whether these structures exist in our bodies is a slightly different discussion. And by structures, I mean the small little particles that can be observed under electron microscopy. It's up for debate. If you're a true purist, I would say that you're going to lean towards no, there's no in vivo evidence whatsoever. There's no in vitro experiments with any sort of external validity that point towards us having these particles in our bodies. Now, from maybe a more alchemical perspective, which is why I think Dr. Berlando kind of leans towards this side, what we see is a macrocosm reflecting the microcosm. So looking at our macrocosmic selves, right, and our cells, as we might know them in a less traditional way, not necessarily the cell membrane, but that we are organized in these smaller substructures and that we continuously get smaller and smaller in our substructures. It's not absurd to claim that these might be extracellular debris, right, that they might be pleomorphic debris in some capacity. The real problem lies in what do we really understand about these particles? And the answer is nothing, right? Because all of our methods fail. They cannot be isolated, so we cannot study them empirically. All we have is a little bit of logic to go off. So I'm not going to sit here and say that I disagree with Dr. Lando. I'm not going to say that. Just at this point, I think that there's just very little evidence to go off of other than pure logic and maybe adhering to some traditional alchemical hermetic principles. So viruses have never been demonstrated to exist. This is a little bit different than saying viruses do not exist because it puts into questions the methods rather than the phenomenon itself. Both are valid statements in my opinion. I know Dennis Rancourt speaks a lot about this and I agree with his points completely. I believe that it can be harmful putting out this message that viruses do not exist into the scientific community if we're trying to foster some sort of scientific discussion. So virology really relies on four major methods. I call them the four horsemen of virology. That relies on electron microscopy, serology, genomics, as well as the cell culture. The reason why it's only these four methods is because contagion experiments failed. Human experiments failed. We've tried to isolate viruses, which we've never been able to isolate, and that's admitted now in the literature. They cannot be purified. They use the term isolation to say, have it in a cell culture, which is not a purification at all. But regardless, all of our in vivo contagion experiments have all failed. So we've moved into the test tube because in real life, instead of accepting our falsification like a true Popperian empiricist would, well, we've decided let's just continue to perpetuate this lie. As Kuhn so gracefully pointed out, the theory-latedness takes over and we cannot accept the falsification. So we've just tried to push it into the test tube to use our unbelievably unscientific methods, circular reasoning, confirmation bias, which we'll be able to see in this video. So obviously the starting point here is isolation. Virologists will tell you that we have isolated viruses, but this couldn't be further from the truth. You see, they've redefined, right, created a new definition for isolation in virology. They've taken away the aspect of purification. So let's think of isolation, right? A true isolation, a true purification is separating a substance from all other parts. So my dog, I can isolate him, I can take him, I can put him in a room somewhere, and this would be a purification. It would just be my dog, you know, like you can purify this jacket, or you could purify, you know, a lake water, right? So you could take that lake water and purify it. Now, if you wanted to purify so-called H2O, you would have to distill it, right? And so when you have 100% H2O, that would be a purified water. Obviously, the idea of contaminants is important in science because of confounding variables, right? If there are a ton of toxins in a sample and they're so-called viruses, and we give that to a human being or an animal and it causes disease, who's to say that it's the virus and not the toxin? Especially when you control a toxin and you realize that both groups are having the same reactions. A quick point I want to make here is that in injection studies, what happens is we give one group a so-called virus or viral particle or attenuated virus or a piece of a virus. We inject one group with that, but our injections contain a whole plethora of ingredients like aluminum, for example. And our control group actually contains the exact same injection, all of these ingredients, which are known to be toxic. Aluminum, for example, is just the most clear example to give here, but there's other things that cause inflammation and disease. So what happens is both groups are receiving the toxin, and so both groups are having the same number of adverse effects. And these studies are in the literature, and you can see clearly that both groups are having pretty high numbers of adverse effects as well. But the so-called viral particle is what's being controlled here, but the toxins aren't being controlled. And there's also no safety studies on these toxic injections either, right? But then they'll say it's safe and effective, and it's total nonsense. And that's kind of a different topic, but I just wanted to point that out, right? The use of adjuvants has always been a thing in virology. We've always had to inject people with the so-called viral particle and some sort of toxicity to ensure that they had some sort of immune response. So what does a virologist think isolation is? Well, it's removing the virus from a person and putting it in a cell culture, right? So we've purified the virus by taking it out of someone and putting it into a cell culture. The cell cultures contain all sorts of additives, right? pH stabilizers. Most notably, they contain antibiotics, antimycotics, right? Because you don't want to have bacteria or fungus growth. And the other notable point is that they actually starve the cell cultures, which we'll talk about in a moment. The other point is that you can't know if a virus has been isolated in a culture without the other methods. So actually seeing the virus in the culture requires electron microscopy, and it requires identification through PCR and serology, right? So the use of antibodies. But the other thing that they'll say is cytopathic effects, right? They'll say that the cell culture is breaking down due to the so-called virus being in the sample. But this comes to the point of toxicity and starvation, the two true causes of disease as we understand in the terrain paradigm. Toxicity being the use of antimycotics and antibiotics in the cell culture, and I'm sure there's contributing factors like the pH stabilizers, and often this isn't discussed in the methods itself, but they actually reduce the bovine serum or nutrient source from 10% to 2%, right? So they starve the actual cells, and there have been controls for this, and they've demonstrated cytopathic effects controlling starving the cells, and that's really the driving factor for cytopathic effects. The cell culture breaks down when it doesn't have food, not because there's a so-called virus present. Jamie Andrews has done this. Stefan Lanka originally was the one who did this. Very simply demonstrated, falsified. The cell culture's absolutely been falsified. It's nonsense. It's useless, especially when we're starving the media, right? So that's the confounding variable. So they don't actually control the variables. They don't put them under the same conditions. They don't starve the mock infection. And when you do starve the mock infection, you get cytopathic effects. And this is admitted again in the literature. Many virologists have admitted that starving the cell causes cytopathic effects. Many scientists and virologists and molecular biologists have come out in these debates that there have been and say there are multiple things that contribute to cytopathic effects, to cell death. It's not just viruses. But how do you determine whether or not it was the virus, right? You need to control that variable. And it's just never been demonstrated empirically. It's been quite the opposite, it's been falsified. So CPE is just cellular damage. It's not proof of existence of a virus. It's not proof that a virus is a self-replicating particle that causes disease. And now we get into the circular aspect of this, right? We need electron microscopy, genomic methods like PCR and serology to confirm whether or not the virus is present, because no one can see it with the naked eye. So let us begin with electron microscopy. Virologists claim that electron microscopy is unequivocal proof that viruses exist, right? They set up a cell culture, they take a picture of it. But the process of electron microscopy is quite extensive and goes uncontrolled, and quite literally is impossible to be controlled. Freezing, dehydrating, staining with heavy metals, rehydrating with a toxic substance, like a glycerol type gel substance to rehydrate artificially the sample, cutting the tissue very, very thin, putting it in a vacuum and beaming it with an extremely high amount of electrons, the light. So there's all of these methods involved in electron microscopy that are necessary for preparing the sample. So obviously electron microscopy shows only dead tissue, dead and altered tissue, right? The use of staining alters the samples, right? Cutting it very thin alters the samples. There's no controls. You cannot control this factor. And you might say, oh, well, the mock infection doesn't show any of these little small particles. Okay, so in the mock infection that was never starved and was never, introduced with the same toxicities, doesn't show these little particles that are the cellular debris, as Dr. Landau would say. And I think that's a valid point, right? When you starve the cells, when you cause disease to the cells, not because of viruses, but because of toxicities and deficiencies, what you get are these little particles in the sample. Now, an interesting observation. Go look at people with lung cancer, kidney cancer, cancers of all sorts, diseases, different types of diseases. Look at electron microscopy samples of their tissues. You'll see these little particles, right? Again, something that's absolutely admitted in the literature is that we cannot distinguish between non-viral-like particles and viruses, so-called viruses. So if we cannot distinguish using pictures by themselves, because non-viral particles, extracellular vesicles, cannot be distinguished using electron microscopy alone, what do we rely on to confirm our findings? Well, we rely on PCR and we rely on serology. And the circle continues. So we have cell culture that's never set an actual foundation for a virus causing disease. Then we take pictures to try to prove that there's viruses in the samples, but there's also viruses in so-called viruses, or pictures of viral particles in any diseased environment in the body. So now we go to PCR, right? And the circle continues. Since the beginning, PCR has been a fraudulent way to garner results. It should not be used outside of the laboratory. You can check out last week's video with Dr. Yunea Tomzik, where we really dive into PCR and dismantle it completely. But PCR really has very little external validity. It works very poorly in complex samples. It may work fine in a laboratory setting under really strict controlled environments, but again, go watch that video. The problem here lies into a smaller circle, right? So viruses have never been isolated, so the genome of viruses have never been isolated. So actually trying to get the genome sequenced requires metagenomic sequencing, computer sequencing. There's no gold standard. We can't confirm the sequence because we've never had an isolated genome. So we take genomes of supposedly millions of viruses, bacteria, fungus, cell cultures, right? There's all these genomic contaminations. We pump it all into a computer. Boom, it pumps out this sequence that's just totally based on probability and our theory latedness that we've encoded into these computers. And we get an assembled genome in silico, right? No Sanger sequencing as a gold standard because we cannot isolate the genome. And what we do with these constructed genomes, because it's all based on small little base reads, right? So there's little, sequences that attach to each other, and it's all based on probability. It's totally nonsense when you really dive into it. You could probably go listen to the Jamie Andrews podcast on that, or Tom Cowan, or Dr. Bailey's. We've talked a little bit more about genomics and metagenomic sequencing. I'll link a blog post down below where we go in depth too. But what we have is this unconfirmed genomic sequence, which we build little primers off of. And then we use those primers to confirm the genetic sequence presence in a sample. So again, a circle, right? You can't avoid this circle. So we sequence the genome, which is unconfirmed, create, a PCR test based off this unconfirmed sequence, and then confirm the sequence with the PCR that's been confirmed by the sequence that we've created out of thin air. That's confirmed. You see, it just goes in a big circle, right? So, and that's the PCR test. So again, never been confirmed. How do we confirm that the PCR test has any sort of validity? Well, what we do is we use serology. We use antibodies to try to prove that our PCR tests are valid. We say, well, we're going to build an antibody test, and we're going to demonstrate that PCR is valid because we're going to do it side by side with antibodies and our serological assays. So antibodies, the most unreliable thing in science, the cause of the reproducibility crisis, the biggest fraud and lie that we continue to tell ourselves, even though there's no empirical data to support it. The lie is that antibodies are specific and they can test for specific things. The reality of the situation is antibodies cross-react. The most easy example to think about is the COVID tests, right? You had the PCR tests and you had the antibody tests, or the antigen test, which use antibody technology. PCR test was totally dependent on the number of cycles being ran, the noise being canceled out, and we knew that we were running the PCR at a much higher cycle threshold than we should have been, 45 cycles rather than the acceptable 33, which still doesn't garner absolutely perfect results. No PCR test will. There's no external validity to PCR. Again, go watch those videos if you want to dive into PCR. And with the antigen test, what you saw is that anybody who displayed any sort of illness, especially flu-like illness, or any sort of lung, or even cancers, or so-called HIVs, all of these things tested positive for the antigen test. And that's because antibodies are non-specific. When you have antibody levels that are high in your body, antigen tests are going to test positive. That's why when we have HIV tests, if you're pregnant, you can test positive for HIV, because when you're pregnant, you have all these antibodies in your bloodstream, supposedly. So antibodies are not specific. They cannot distinguish between viruses themselves, and this is admitted in the literature. If you look up the pox viruses problems with antibodies, they say that antibodies cross-react across all the pox families because they're so similar in sequence, which is, again, something we could dive way deep into. But it's totally, unfalsifiable statement. It's never been validated at all. It's totally based off confirmation bias. It's based on our theory-ladenness, and it uses circular, unfalsifiable methods. But what happens is that even in the injection vaccinology field, they admit that one pox injection can have cross-protective measures, right? So you can even think back to the old Jenner days, Edward Jenner, when he, injected that boy with cowpox, and it protected him against smallpox, right? So why is there cross-reactivity of those antibodies and not the antibodies that we use to determine whether or not there is a so-called virus in a sample? Right? So it's unspecific, it's non-specific. Every single antibody cross-reacts. You can go watch this video where we kind of dive deeper into the use of antibodies, but they're non-specific. So how do we confirm, suppose they are specific. Let's say antibodies are specific. How do we confirm an antibody test? Well, we use PCR, or we confirm our samples with electron microscopy, or we use cell cultures and cytopathic effect studies. But aren't we confirming those methods with the serology method? Aren't we, PCR, serology, electron microscopy, cell culture, PCR? So one big circle. Again, a big circle. It's a huge circle. It's confusing. Not many people, scientists especially, they don't see it because of how large the circle is. And that's a big problem. It's a really big problem. People don't see this. But it's circular reasoning nevertheless, right? There is no foundation. There is no, look, here is the virus, this is our foundation, this is our starting point, this is our fundamental premise. No, it always comes back to the premise being affirmed by the conclusion, right? Cell cultures and cytopathic effect studies are valid because we've confirmed them using electron microscopy. But electron microscopy is validated because we use PCR, and PCR is validated because we've used serology. And serology is validated, well, because look, the cells are dying in the cell culture, and we could see them in electron microscopy. And look, these correlate perfectly with PCR, even though it takes so many, there's no study actually out there that does side-by-side PCR and serology to confirm each other's results. It just has never happened and never could happen, right? Look at the COVID example again. PCR positive did not equate to an antigen test being positive. These were rarely ever side-by-side positive. You could go and they admitted that the antigen tests were unreliable. Those didn't count to a case, right? If you tested positive for an antigen test, you had to go get a PCR test to confirm it, right? And you'd test positive and then boom, you'd test negative PCR, right? It was just this total nonsense, right? So we mentioned earlier that we had to go into the test tube because our contagion experiments have just failed. The first studies that I looked at were the Rosnow experiments, where they really controlled natural methods of transmissibility. They also actually injected people with supposedly, virus containing mucus or cough up or whatever, right? Like they literally took that and they injected it, they put it in people's nostrils and their throats, like totally unnatural ways of trying to produce transmission. No one got sick, right? There are over 200 examples of this in the literature, especially when it comes to viruses, right? Viral transmission, it's just never been proven. Even supposedly sexually transmitted diseases have never been proven. There's been super unethical Guatemala syphilis studies where they've tested syphilis and gonorrhea with prisoners and prostitutes who were displaying syphilis symptoms. None of the prisoners got sick, even though they were forcing them to have sex. And they actually went as far as to abrade the ends of the prisoners' penises to try to make the transmission easier. They were bleeding and having sex. It was the totally unethical, terrible experiments. Nobody got sick, right? Transmission, contagion of a viral particle has never been established. Real contagion is the contagion of our environments, when we are in environments that share toxicity or when we're in environments that are deficient. That's where we get sick. So the whole idea of asymptomatic carriers was sort of this backfill that we've been discussing a lot recently. Totally illogical, goes against Koch's postulates, which is a whole other story. But again, the idea that there can be an asymptomatic carrier is just so that we can explain when someone tests positive with PCR and they're not sick. We say, well, they're an asymptomatic carrier, but they're still contagious, right? That idea perpetuates the transmission lie where we say, we can track cases. Well, it's because if you test enough people will test positive and they don't even have to be sick to be a case, right? So you could prove a contagion of absolutely anything using that model if you have everyone in the world testing like millions and millions of times, probably billions of tests. The idea of a strong immune system is a totally unfalsifiable claim as well to explain out this asymptomatic carrier. It's just fundamentally illogical. It makes absolutely no sense. So we really have to move past that. It's just backfill, right? It's just backfill to try and explain out why our observations and methods are failing and not proving our theory, right? But it should be a falsification. So everyone would probably go, oh, well, what about, polio and what about all these, sexually transmitted diseases? Is just, HIV is kind of the easy starting point. Go and look at those studies. It's just that, it's not that people don't get sick. People get AIDS and people get, polio and people get all of these different illnesses, syphilis, gonorrhea. People get the symptoms, but it's just not caused by a virus, right? And this is simply demonstrated in the literature. There's no valid methods of proving viruses to even exist, but there's just no correlation between virus presence and the disease itself, right? Asymptomatic carriers are false negatives, or it's all just backfill to explain this stuff. So what does cause disease? I said it earlier, toxicities, deficiencies, traumas, right? So physical or maybe psychological traumas can cause some sort of disease indirectly or directly. We'll talk a little bit about that more in the community for sure. And how to implement this into your life. How do we, live in a life in a modern world that's so burdened with toxicity that we're all deficient, and we're all living in these deficient environments spiritually, mentally, physically. These are the true causes of disease. Microbes, viruses, cholesterol, hormones, none of these things cause disease. It's very, very simple. They've just never been proven empirically because they cannot. They've all been falsified, really. So that concludes our episode. Go read the blog post down below if you want to dive more in depth. I have some great podcasts on falsifying virology as well. The most recent upload with Dr. Yanea Tomzik is fantastic. Sun is finally coming up, so I'm going to enjoy morning with the dog here, but I just wanted to pump this video real quick. Virology is an absolute pseudoscience. It's been falsified. We cannot rely on it whatsoever. It's just absolutely useless field, among a couple others. At least microbiology studies real phenomenon, but again, we still don't understand nothing about microbiology. Science cannot escape logic. We cannot just say that we're confirming our results when we're stuck in a circular reasoning, when our methods are circular. We can't just say that it's confirmatory, right? It's confirmation bias. We're saying that it's being confirmed because that's the results that we want. It's theory-ladenness, right? It's total nonsense. We can't just be illogical in the name of science, because we want these theories to be real. We don't have real-world examples, and we don't even have laboratory methods to prove this positive. So we need to completely reject the hypothesis and the theory and move past it in a more meaningful manner. And really, that's what the Beyond Training community is about. So go over there and join the waitlist. We're going to be moving forward away from all these nonsense methods and terrible theories and ideas. We're going to raise healthy children. We're going to be happy. We're going to have a great community, online community, have real world communities. We're building something really, really cool. This is just the start. You're going to want to be in the community, not just for the community itself, but for the vision of Beyond Terrain. It is going to be a lot bigger than just the community, but get in that community because you'll be able to shape it. Be able to shape this vision forward. I want to work with all you guys, for all you guys. I really love this stuff. So go over there, join the community. We'll be able to build something together, right? Do some cool things. So hope you guys enjoyed the video. Really appreciate your time. Take care.